people think about blood, they think of something painful or unpleasant that may have happened, like an injury. And in fact, many people have an aversion to blood. But blood, as you'll hopefully come to see during this tutorial, is a fascinating substance, if not only because it is so complex that we're still discovering new things about its composition, but also because it's so vital for life. Blood has fascinated us for millennia. In ancient Greece, blood was known as haema and was considered synonymous with life. To visualize the cellular components of blood using light microscopy, a simple preparation known as a blood smear or blood film is used. Here we see a micrograph showing a blood smear, and as you can see, several cells can be seen and distinguished. Blood is composed of cells and cell fragments suspended in an aqueous solution called plasma. This technique uses a single drop of blood which is placed onto a glass slide. The drop is spread thinly over the surface of the slide to produce a single layer of cells. The sample is then allowed to air dry. In order to visualize the cells, a stain or a dye is added. These stains usually contain methylene blue or other azure compounds, which are basic dyes, and eosin, which is an acidic dye. In this slide, you can see how the stain makes the blood cells different colors. For example, the light purple of the red blood cells and the dark purple of the lymphocytes. These little purple dots are platelets. The different compounds bind to different parts of the cells, allowing them to be distinguished from one another. The stain is applied to the dried blood smear for a few minutes, then rinsed off. Giemsa and Wright stains are two common stains used for blood smear preparations. Our image today is of a Wright stain, which is made up of a combination of methylene blue and eosin. Wright stain is used to show blood cells and sometimes malarial parasites. Blood plasma, which is the aqueous solution in which the cellular components of blood are suspended, is itself comprised of the proteins albumin, alpha, beta, and gamma, globulins, fibrinogens, and other complement proteins. In this slide, of course, the blood plasma is transparent and is therefore the white spaces you see in the image. Let's talk briefly about what we're going to be covering in this tutorial. In this tutorial, we'll be focusing on the cellular components of blood, starting off by looking at the erythrocytes, or the red blood cells themselves, followed by the leukocytes, or the white blood cells, and finally we'll look at the blood platelets, which are another important component of blood. So without further ado, let's get started by looking at erythrocytes. Erythrocytes, more commonly known as red blood cells or RBCs, comprise about 45% of the total volume of blood. They're of course the cells you can see highlighted in green. The percentage of red blood cells in the blood is known as the hematocrit. As we can see from this little diagram here, the red blood cells themselves do not contain nuclei and are biconcave in shape. They measure about 7.5 micrometers in diameter, about 2.6 micrometers at the rim or edge, and only 0.75 micrometers in the center. Their primary function is the transportation of oxygen to tissues and carbon dioxide away from the tissues. What allows this is the fact that red blood cells are filled with the protein hemoglobin, which is an oxygen-carrying protein. In fact, the entire cytoplasm of red blood cells is filled with hemoglobin, which reversibly binds oxygen and carbon dioxide, allowing the red blood cells to transport these gases from the lungs to the tissues and vice versa for gaseous exchange. The cytoplasm of red blood cells does not only lack a nucleus, but also lacks organelles. And just to remind you in case you've forgotten, an organelle is an organized or special structure that performs a particular function in a cell, like a little organ. The lack of organelles in a red blood cell allows more hemoglobin to be packed into the cytoplasm, and therefore more oxygen to be transported. The cells lose their nucleus and organelles during differentiation in the bone marrow, shortly before they're released into the general circulation. Whilst we're on that point, let's take a brief look at the precursor to red blood cells, known as reticulocytes, some of which we can see in the micrograph here. Reticulocytes are immature red blood cells, which develop in the bone marrow before being released into the circulating blood, where after 1 or 2 days, they eventually develop into mature red blood cells. In healthy individuals, reticulocytes account for only 1 or 2% of the cells in blood. However, this number increases in instances where the demand for oxygen outweighs the supply, in which case reticulocytes are released from the bone marrow early before they mature into erythrocytes in order to increase the blood's oxygen-carrying capacity. As such, reticulocyte counts can be used as a marker for anemia and to check for bone marrow disorders. Moving back to our more mature erythrocytes, their structure means that they're very flexible, and this allows them to easily deform or bend in order to pass through the narrow vessels of the capillary bed. Glycoproteins and glycolipids, both of which are kinds of antigens, are present on the surface of red blood cells. There are 3 types of antigens, which differ slightly in their composition and are denoted by the letters A, B, or O. The presence or absence of these antigens in an individual is what determines that person's blood type. For example, people with type A antigens on the surface of their red blood cells are described as having type A blood, and so on. The presence of these antigens and knowing them is clinically relevant with respect to blood transfusion and blood type compatibility. The lifespan of red blood cells in circulating blood is about 120 days. When red blood cells become old, they lose their ability to deform, which is detected by specialized cells in the spleen. The spleen extracts them from the blood. breaks them down and sends the components to the liver for recycling. Now let's look at another group of cells that make up the blood, known as leukocytes. Leukocytes are more commonly known as white blood cells, or WBCs. The word leukocyte is derived from the Greek word leukos, meaning white or clear, and cyte, meaning cell. These cells generally have a lifespan of around 120 days. In this slide, do note that we're looking at a leukocyte under an electron microscope, as opposed to a light microscope, as evidenced by these bright purple leukocytes you can see here. There are 5 types of leukocytes, which we'll be covering in this section of the tutorial. These are neutrophils, eosinophils, and basophils, as well as lymphocytes and monocytes. These can be classified into 2 different categories. Granulocytes contain specific granules in their cytoplasm. Agranulocytes, on the other hand, lack these specific granules in their cytoplasm. However, it's worth noting that both granulocytes and agranulocytes possess non-specific azurophilic granules known as lysosomes. Let's take a look at each type of leukocyte in more detail, starting with the neutrophils. In this image, note that we're looking at an artist's visualization of a neutrophil very close up, as we can't see them with the naked eye. As we just saw, neutrophils belong to a granulocyte group of white blood cells. They range between 12 and 15 micrometers in diameter and make up between 54 to 62% of circulating white blood cells. As you can see in this micrograph, the nucleus of a neutrophil is lobed and typically exhibits 3 to 5 lobes. That's why in some literature, you may come across neutrophils being referred to as polymorphonuclear leukocytes, or PMNs. These nuclear lobes are linked together by thin nuclear extensions. In females, the inactive X chromosome appears as a drumstick-looking appendage. On one of these lobes, which we can see in our image here, the appendage is known as a bar body. It may not always be visible, but it's worth being aware of. Neutrophils belong to the granulocyte group of white blood cells because they contain specific granules in their cytoplasm that stain a light pink after hematoxylin and eosin staining. These specific granules secrete ECM-degrading enzymes, which are enzymes that degrade the extracellular matrix. And the granules are also what provide neutrophils with their functional phagocytic capability. You can think of neutrophils like first responders. They're the first to be attracted to and arrive at the site of infection via chemical mediators, moving to the site through a process known as chemotaxis. As phagocytes, neutrophils engulf bacterial cells and other small particles. They are key players in the inflammatory response. Neutrophils release polypeptide chemokines and lipid mediators of inflammation. The release of polypeptide chemokines acts to attract other leukocytes and cytokines that direct the activity of neutrophils and other cells involved in the immune response. The lifespan of a neutrophil is short, being only around 1 to 4 days. The next type of leukocyte we'll look at are the eosinophils. Again, we're looking at an artist's impression. Eosinophils comprise about 1 to 3% of leukocytes and are roughly the same as neutrophils. These are also granulocytes and develop and mature within bone marrow, after which they're released into the peripheral bloodstream. Here in this micrograph, we see an eosinophil in a blood smear. As you can see, this cell type of granulocyte has a bilobed nucleus, and like the other granulocytes, it contains oval granules in its cytoplasm. In addition to these, eosinophils also contain other substances such as major basic proteins, or MPPs, eosinophilic peroxidases, and enzymes, and other toxins that act to kill helminths and parasitic worms. Eosinophils can be found in abundance at sites where chronic inflammation occurs, such as the lungs and in the connective tissue of the intestinal lining. This is because eosinophils, among their other functions, modulate inflammatory response. Eosinophils are triggered into action by allergies, and they release chemicals such as cytokines, lipid mediators, and chemokines. The third type of leukocyte we have are basophils, which are also granulocytes, and which actually make up less than 1% of leukocytes. They are 12 to 15 micrometers in diameter and contain specific granules that stain purple or basic on a blood smear. Here we return to our micrograph of a blood smear, with a basophil highlighted here in green. The granules in basophils stain so highly because they contain heparin and other sulfated glycosaminoglycans. In addition to this, the granules present in basophils also contain histamine, pro-inflammatory factors called leukotrienes, and other mediators of inflammation, which are released in response to certain allergies and antigens. Now that we've looked at the granulocytes, let's look at the leukocytes that are classed as agranulocytes, starting with the lymphocytes. Lymphocytes are the second most abundant leukocytes found in blood, constituting around 25 to 33% of all leukocytes, and they're typically the smallest of all the leukocytes. They possess spherical nuclei, which we can see here in this micrograph of a lymphocyte. As you can see here, at the center of the leukocyte is a round nucleus with only very minimal cytoplasm. Lymphocytes can be subdivided into groups based on the surface molecules they possess, known as cluster of differentiation, or CD markers, as you may know them. These CD markers, which can be distinguished through immunocytochemistry using antibodies, subdivide lymphocytes into 4 major classes: B lymphocytes, helper T lymphocytes, cytotoxic T lymphocytes, and a natural killer or NK cells, all of which play a number of roles in immune defense against parasites, microorganisms, and abnormal cells. The lifespan of lymphocytes varies according to their specific function, and they can survive in circulating blood anywhere from a few days to several years in certain tissues. Now let's take a look at the other type of agranulocyte and the final type of leukocyte we'll be looking at in this tutorial, known as monocytes. Monocytes are typically 12 to 15 micrometers in diameter and possess a C-shaped nucleus. Their cytoplasm stains basophilic, as you can see in this micrograph, and contains small azurophilic granules called lysosomes. The high distribution of lysosomes is what causes the cytoplasm of monocytes to appear bluish in colour, as you can see in this micrograph. It should be noted that monocytes are precursor cells of the mononuclear phagocyte system, including osteoclasts, microglia, and macrophages, like the one seen here in this micrograph. All monocyte-derived cells are antigen-presenting cells, and as such play an important role in the body's immune defences. Now let's take a look at another important component of blood, which are the platelets. These non-nucleated cells measure between 2 to 4 micrometers and function to promote blood clotting. Platelet cells also play a role in tissue repair, such as the repair of minor tears and leaks in the walls of blood vessels. Here in this micrograph, we see platelets, which are also known as thrombocytes. They are in fact fragments of the phospholipid membrane of a larger cell called a megakaryocyte found in the bone marrow, which has released them into the bloodstream. In blood, the normal count of platelets or thrombocytes amounts between 150,000 to 400,000 per microliter, and they have a typical lifespan of up to 10 days. Now that we've had a look at the components of blood, let's briefly cover a blood-borne disease that affects millions of people and results in hundreds of thousands of deaths every year, which is malaria. Malaria is an infectious disease transmitted by infected female Anopheles mosquitoes to humans and to other animals. A bite from an infected female mosquito introduces the parasite Plasmodium from the mosquito's saliva into the bloodstream, which we can see in this magnification here. These parasites cause malaria symptoms, occurring within 10 to 15 days after infection, which include fever, vomiting, headaches, and fatigue. In several cases, this disease can lead to death. Malaria can be diagnosed through the microscopic examination of blood films, such as the one seen here, or by antigen-based rapid diagnostic tests. Blood smears or blood films like the one seen here detect the parasite using light microscopy and specific staining techniques. To treat this disease, medications are prescribed such as chloroquine, mefloquine, or doxycycline to kill the plasmodium in the blood. This brings us to the end.